Good morning and welcome to CitraChem's Cannabinoids, a new generation of medicinal opportunities. It is 9:30 a.m., the start of our program. I'm sure people will be logging in. So I thought I would just start with some of the logistics and then I'll get into some background, talk about the agenda. The logistics are this is a virtual event. Everybody attending can see and hear slides and hear the presenters. If you have questions, please use the Q&A functionality in Zoom. We will have someone monitoring the Q&A. So after each presenter, we can take some questions if we have time. We'll be mindful of the time to keep everybody into the schedule that we've set up. We're really excited about presenting today. This is a bold new area of medicine, and I think our takeaway already from our aspect of the business is how exciting it will be to find new ways to use plant-based molecules, cannabinoids in this case, to address a lot of unmet medical needs. So CitraChem is really at the heart of a lot of what you'll see in the presentations, but each presenter will have significant resources that they've been putting on these topics that they're going to present to you today. So we hope you appreciate that this is already moving forward. Part of the discussion today will be how the US government has put a bigger spotlight on using cannabinoids to research and find new medications. So that's the premise of today. We're really glad that you joined us. Let me go through the agenda. Again, while we expect more people will join us, let's just talk about what we're setting up for today. So I'm going to go through a background about CitraChem, overview of the premise of our company. Following me is Jeff Schultz from Foley Hoag, who can give you a brief background on how the government has treated cannabis molecules in the past and what this new dynamic should result in for medicinal research based on rescheduling cannabis. Dr. Yossi Tam will follow. He's at Hebrew University, one of the premier cannabinoid research scientists in the world. He's got a lot of great topics he could cover, but in the time that we're given, he'll give you a brief overview of his work. That will be followed by Dr. Jason Kingsbury, who is CitraChem's co-founder, inventor, chief scientific officer. So Jason will go more into exactly what our platform does and how we contribute active pharmaceutical ingredients to the research projects that you're going to hear about. And then starting with Dr. Mark Chaldone at 11, Mark's a professor at St. Michael's College doing research work for CitraChem, but has his own cannabinoid research experience. So we'll start to hear with Mark the essence of how we go about helping those who are doing cannabinoid research. That will be followed by Jeff Hareen, who has a company that has been involved with cannabinoid products for a while. And he's going to talk about their special patented formulation, which is really interesting. Something that we talk a lot about. How do drug discovery companies and drug companies benefit from protection with patents? And then last, we'll have Dr. Barry Berkowitz. He is here in our corporate headquarters where we're talking to you now. And Barry will talk about how you go about a real FDA application using cannabinoids as the active ingredients. So what we're hoping to demonstrate here is there's real science going on. There is significant effort and resources being put into adopting cannabinoids in various forms to make medicines. And we do believe in others, the presenters and other clients, there's real benefit to using cannabinoids, combinations, et cetera, to address these unmet medical needs. Before we turn to my content, I'm just going to check in. Looks like we're getting a good number of attendees coming in. So I think we will start with the presentations. So I am James Winokur, the CEO of CitraChem. We are a company that has a chemistry platform to produce botanically identical cannabinoids, exactly the same as the plant can derive and or we humans have some kind of effort to transform them, which is also what you typically see in the cannabis world. but we also can make derivatives. So what we'd like to do today is really emphasize using nature to transform molecules that can be to the benefit of what we consider typical FDA processes to derive acceptable medicines. So when we think about what's going on in the medical world, CitraChem knows from evidence that we've researched and certainly others have brought to us, there's a broad spectrum of diseases, conditions, and ailments where cannabinoid medicine could be applied. So there are hundreds of molecules in the cannabis plant. Sometimes I call it a treasure trove of molecules that already exist. Some of them are abundant in the plant, some of them are not abundant at all. And if you take that 150 or so cannabinoids or 500 chemicals in the plant, we have a lot of research that could be done to find the efficacy and the safety in many, if not all of these molecules that could benefit the diseases on the screen and many more that are not listed. So when we think about the work that you're going to hear from Dr. Kingsbury in a little bit, it's how do we use a platform to make as many of these molecules as we possibly can. And you'll hear that we've already proven out our platform. Started with proof of concept, but we've now moved to 3 molecules at scale, which means they can be GMP and they can be produced commercially. Even though there's small amounts that will be used for research, we've already proven out that we can do this at a pharma-grade and a volume needed. So if we typically look at a molecule under microscope on the far left, Dr. Kingsbury would look at that and say, yep, I can replicate that. But we also have clients who say, well, what if you could make a variant of that? So the molecule shown under the microscope might be close to the original, but if a client wanted to derive composition of matter claims, i.e. a patent, we can use our same platform to make that happen and get a derivative. And then we expect that will go into research and then eventually commercial drug. So what we are offering is the ability to have a consistent molecule at pharma grade at the volume that's needed. And some of our clients have said, if we are providing that right from the beginning of the early stage discovery, it strengthens their drug application in the future to have a source that can provide exactly what's needed at commercialization. That's our goal. So if you want to think about an analogy, CitraChem is providing this active pharmaceutical ingredient library of botanically identical molecules. We're doing custom work to create derivatives. That makes us like Intel. We're inside all of these medicines. We are not a drug discovery company. We're supporting, empowering the drug discovery companies that need these APIs, the active pharmaceutical ingredients. And we are really not focused on a single condition or a group of conditions or ailments. We are really kind of agnostic to that. So when clients come to us and they have a target disease, we want to help them by finding the right molecules for their project, their program. And in fact, if you think about it, we can do that for hundreds of medicines, hundreds of companies that want to do drug discovery. So that really takes us out of the single or maybe 2 or 3 target diseases to we want to help as many companies as we possibly can. Now, there is a confluence of things happening right now in the world. One of the issues that pharmaceuticals are now concerned about is what they're calling the patent cliff, where they have a number of drugs that are protected. When they come off patent, they go to generic. So the numbers on the right are to demonstrate the revenue at risk of a lot of drug companies, which means they're in a mode of trying to backfill what they can produce as patented medicines. And they want to do that as quickly as possible, as you can imagine. So there is a movement right now to how do we find new medicines that we can patent? At the same time, we have learned that when the FDA drug applications are submitted, there's a good portion that are completely novel molecules, more than the majority. But when they're completely novel, they might lack some safety ability. So the success rate of a completely novel molecule is only about 10% getting approved by the FDA. However, if you start with a compound that is actually based on a natural, as I said, we can replicate or we can make a variant. If it's based on a natural, it has a 45% success rate. And that's really where we want to help. So if you combine these things, pharma companies are looking for new patentable compounds, and we can produce compounds based on naturals that might be a variant, so you can get those composition of matter claims. We're kind of in a sweet spot right now as CitraChem to help provide those. One really interesting aspect of what we're doing and what our clients are doing is the different variations of how you can use these molecules. So as you're listening to the other presenters, I'd like you to think about this slide a little bit. You know, the pill bottles here in a clock form. So in the middle, you see identical acid form. So a cannabinoid from the plant starts as an acid form. And we know that we can replicate those, and they may have very good medical benefit. If you go to 12 o'clock, that's typically what you're probably aware of. So a molecule like CBD or THC or CBG, we hear those all the time. They're actually transformed from the original acid form, but those are the ones that we're well aware of and a lot of studies center around. So some of the presenters today you will hear are using molecules in that vein, that they are now in the CBG, CBD type of molecule. But if you go to the left at about 11 o'clock, you'll hear at least one of the presenters talk about, well, what if we use more than one molecule together, a co-therapy, so that the combination in that formula is more beneficial than any single molecule on its own. And if you go to the right at about 2 o'clock, 1 o'clock, it says synergist. There's also the idea that you can take a cannabinoid molecule and pair it with a non-cannabinoid drug. We've seen this, for example, with Epidiolex, which is a Jazz Pharmaceutical FDA-approved drug. It's a CBD-based drug, but it is often actually paired with a non-cannabinoid-based drug, and the two of them together seem to be more effective. So that's what we mean by synergist. And then down at the bottom, derivatives, that is the idea I just mentioned, which what if we were to take a CBD and make different versions of it that might be more beneficial and derive patent versions. So just take a quick look at this slide and understand as you're going through listening to the presenters, they represent a lot of these categories. So it's not just one thing. How can we use CBD or CBG? It's actually multiple ways that these molecules can be used. And that's what's pretty exciting for us. We are excited, but I think the world's excited. Dr. Sanjay Gupta has been offering a series on CNN for many years now called Weed. So he's been following this for a long time. He had an episode called Weed 6 out earlier, and he made this statement that he thinks there's a huge development here, that the United States has put a stamp by rescheduling cannabis from Schedule 1, no medicinal value, to Schedule 3, there is medicinal value. That will really open up a lot of research. And when we hear from Jeff Schultz in a minute, he'll talk a lot about that. So what kind of business is this? Well, Jazz Pharma is kind of the poster child for what could be. GW Pharma created that Epidiolex drug I mentioned. Jazz Pharma eventually acquired GW for about $7.2 billion. So they looked at the opportunity for a drug with a very specific condition in mind, and that was a big bet for them. But that's kind of the poster child of what could be just for that one drug, one molecule, really. And then recently, Rolling Stone had an author write about what's changing with the federal laws. And if you get a chance to find this Rolling Stone article, you can see the title there. It really presents a great way to understand what CitraChem is doing and what our partners are doing that you're going to hear about, which is to take this, idea of the molecules. We have clues, evidence, and some really good studies way beyond where we've been. And that includes using some of those less abundant molecules to start studying. So if I say to you, hey, we've been contacted, people want to research CBL or CBT, you may never have heard of those molecules, but as I said, there's hundreds of them. It may turn out that something that's not abundant in the plant has a really good medical property. So that is also something CitraChem can provide, those minor, what we call minor molecules in abundance. So that's a big overview for CitraChem. You'll get a little more detail on the science from Jason in a minute. I have my information up there, so please, if you want to know more about CitraChem, contact us. And then as we go through this agenda, we're here in Norwood, what I call world headquarters. A number of the presenters are remote. So we will ask right now Jeff Schultz to log in. I will stop sharing. And Jeff, I'm going to do a little introduction for you, and then we can start with your presentation. And I am trying to stop sharing. I'm just aware I am. Not being cooperative. Okay, there's Jeff. Hello, Jeff. How are you?
Morning.
Morning. Let me give you a nice introduction, and then I'll let you start on your way. Are you able to share your screen?
I can get to the.
I'm seeing your first slide. That might be mine though. I'm not sure if it's mine or yours. I'm looking at a screen over here, so it doesn't look like you're sharing yet.
No, okay. I'll get that set up.
Okay, so while you're doing that, I'll give you your introduction. Jeff Schultz is a partner at Foley Hoag, where he serves as a trusted advisor to cannabis industry operators and investors of all sizes. He helps clients navigate the complex and dynamic regulatory landscape by leveraging the combination of deep industry knowledge and expertise with 18 years of legal practice. He focuses on guiding cannabis operators through capital raising, joint ventures and strategic alliances, state cannabis regulatory matters, corporate governance, day-to-day commercial contract negotiations, and M&A. He also represents private equity funds, hedge funds, debt funds, and family offices on securities law, fund formation, SEC compliance, venture investing, M&A, and cannabis-related regulatory matters, including capital markets and AML issues. So that is Jeff. As far as I can see, Jeff, you're not yet sharing. Are you having an issue sharing? Looks like we're coming up here. I see you're starting to share. There you go. All right. I'm going to turn off my camera, but my audio will be on. So if you have any questions for me, let me know.
Jeff, you're muted.
Sorry, it's my first time on Zoom. Are you only seeing do you see just the slides right now on my screen? Is that what's being shared?
We're seeing your non-presenter mode.
Let's see. Make sure I got this right. You see the presentation though? I just want to make sure
We see the presentation, but it's in your preview mode.
Got it. Let me just make sure I got that. Sorry about that. Let me just make sure I've got this. Presented. Well, I can keep it like this. As long as you can see, you just see the side panels.
Correct. Yeah.
We could skip my bio. Well, if this looks good, if you could see the slides, then let's get started. So thank you for the introduction. It's great to be here. This is really, really exciting. I've had a lot of conversations in the last month with a lot of folks in the industry, and my takeaway is there's a small handful of smart people paying attention to what's going on. There's a lot going on, but there's a lot of smart people that are focused on the medicine here, and the landscape has changed dramatically in the last month, and it feels almost a little bit like the industry is in a little bit of shell shock. It's quite an extreme development. that we're going through right now, and perhaps I think most would agree that this is the biggest paradigm shift we've seen in the cannabis industry, perhaps in the history of the country, and it's the first time that the federal government has acknowledged the cannabis is medicine. On April 23rd, the DOJ moved to legalize medical cannabis, including state licensed medical operators. And they finally moved cannabis off of Schedule 1, which is the same category that heroin is on, to Schedule 3, effective immediately. There's no phase-in, there's no waiting period. State licensed operators still need to apply. They have a 60-day window. They now have 23 days left to apply for a DEA registration and license. That's not a deadline to apply. That's just a deadline right now to get an expedited process through. Query how long it'll take if anybody applies after that date, but anyone who's applying before or on June 26th apparently is guaranteed to have a response, or at least have their application processed within 6 months, which is relatively quick. The second part of that is another ALJ. There's another ALJ hearing that will begin on June 29th. And that's to determine the fate of the rest of cannabis, including adult use. Why should we care about any of this? Because Schedule 1 is not just a legal classification. It's really been a wall, a virtually insurmountable wall, except for a handful of operators. And it's a wall between cannabis and the future of cannabis as a regulated, evidence-based pharmaceutical. And it's, like I said, I cannot overstate the importance of this change, right? We legalized medical cannabis. The doors have swung open. That's a wall that's been up forever. It's a wall primarily from where I sit between science and raising capital and creating medicine for people based on plants. It's a wall that has prevented a large number of operators from acquisitions or takeouts like GW for $7.2 billion, and dozens of companies that are following quickly behind them. And that lack of institutional capital, the massive operational hurdles like banking, insurance, lack of supply chain sophistication and scale, that wall is coming down right now as we speak. It's not tomorrow. It is absolutely today. And this is a bit of a call to action, both for operators and investors alike. Now's the time. Now's the time to be looking at this. And I'll also say, like, this has really helped, mitigate the stigma around the plant. And I think that goes a long way, and it's hard to overstate the importance of that as well. So what does that mean? That means, companies with FDA cleared INDs, phase 2 trials, GMP manufacturing already in place are very much in the pole position, and they're ready for that scale. They're ready for acceleration and to move on and can raise money without a lot of the barriers that have existed. Perhaps not all, but most of them. So what happened? The final order came out on April 28th. What it did was a few different things, right? It scheduled the hearing that's supposed to begin on June 29th. There is a tentative deadline of July 15th, query whether that deadline will be met. Although I will say that although the hearing may end on July 15th, that does not mean that there will be a final determination from the ALJ at that time. They will issue their decision and a rule, and there's a process that will follow that will take many, many months to play out. However, that's all with respect to adult use. As of right now, medical cannabis is legal, and that comes, it stems from early days during the last Trump campaign, um, the president made a lot of promises about this, specifically on medical cannabis, and then followed up with his executive order in December. And he didn't mince words. I don't think that anyone who's reading this saw this. It was very clear. He wanted it done, and he wanted it done quickly, which is great. And that's happening. So what does this all mean? Schedule 1 versus Schedule 3. Schedule 1 drugs have no accepted medical use. They have a high abuse potential, and no accepted safety under medical supervision. I think we all know, as it relates to cannabis, that's an absolute farce, completely ridiculous. Schedule 3 Schedule 3 drugs have an accepted medical use, a low abuse potential, and have moderate to low physical dependence. Makes a lot more sense. What does this mean? Big picture, the federal government has now formally recognized cannabis as a legitimate medicine with legitimate medical applications, based on HHS's findings, of over 30,000 healthcare practitioners. It's a lot. It's an overwhelming number across 43 jurisdictions. Currently recommend cannabis for over 6 million patients across the country. I think it's now just a point where the government has acknowledged what's actually happening in practice. There are a few very minimal FDA-approved products today, including Epidiolex and Marinol. And as I mentioned, Jazz acquired GW for $7.2 billion. Epidiolex is doing $1 billion in annual sales. And this is just, to me, the tip of the iceberg of what we're about to find out and see and discover and hopefully make people feel a lot better. How does it change research? Right. So Schedule 1 substances, doing research of Schedule 1 substances was possible, but extremely difficult. You needed site-specific DEA registration protocol per location, DEA investigator registration for each protocol, and largely there was a single source of supply from with NIDA's agreement with Ole Miss. And I remember watching the Sanjay Gupta. I don't remember which episode it was, but they toured the facility. They showed at least some of the bulk flower, and I'll just say that I wasn't impressed with what I saw on the TV, and that can't be the only source of product. If we're going to take this, if this industry is to be taken seriously and real research is actually going to happen. It's a very, very long timeline for every stage of FDA approval. And you have review boards, and like I said, the stigma has really slowed it down, if not stopped a tremendous amount of that research and progress. What's going to change now, right? Why does this really matter? And what doors really did just get blown open? Well, first and foremost, cultivators across the country with state medical licenses now have that opportunity to apply for and obtain a DEA registration so that they can be a supply source for researchers across the country and internationally. There's a safe harbor that I think is worth mentioning in the final rule, that no adverse action will be taken against any researchers who source their input products from a state licensed medical cannabis company. And that's just going to allow for tremendous scale. With scale comes revenue expansion, particularly for incumbents. The addressable customer base is going to grow exponentially. I don't know if it happens quite overnight, but really quickly, I would imagine it's about to happen. You now have companies who can supply, take CitraChem, for example. CitraChem will be able to apply products and sell products to multi-site researchers across the country. That has not been easy, if possible at all, right? So I just want to mention that not only will there be a lot of new business opportunities that we have yet to see, but I want to emphasize that there's a huge strategic advantage for the first movers in the space, of which there are very few. Move on to. So why was this the case, right? Federal illegality. Look, we've been living it. I've been in the industry for 8 years. I got into the industry as an investor in the space, and just being part of managing a institutional grade private fund that was invested exclusively in the cannabis industry comes with a host of complications that frankly just detract and distract investors from their primary mission of being good stewards of capital. You have all kinds of institutional funds that have restrictions. Of course, there are restrictions in their fund documents that don't allow them to invest in federally illegal industries. That's gone. It's gone overnight. We'll see where we go with adult use, but right now, these companies are all federally illegal. They should be able to open up bank accounts, obtain insurance policies with reasonable premiums, and everything that goes along with it. Eventually, hopefully, we get to see. And let me just mention, just on with respect to capital markets, medical cannabis companies, there does not seem to be any barrier to listing on the NASDAQ or New York Stock Exchange. That's huge. I looked at that as an investor. We always thought investing in private companies in the cannabis industry, the real value unlock was going to be IPO and public market access. And that just has not been the case in the 8 years I've been in the industry. And that will change at least for the moment, for exclusively medical cannabis companies. They don't have that issue. This is very much a traditional standard, biopharma industry now, with no barriers to capital markets. Hopefully, we get to see the same ability of multi-state operators and adult use cannabis operators or mixed adult use medical operators do the same thing in the coming months or years. Yeah, it is no longer technically illegal. It is now completely illegal. Everything that would normally apply from a diligence and investment oversight perspective applies here, right? Diligence is more reliable. There are a lot of variables that have been erased overnight when looking at outcomes. What does this mean for many of the operators, right? Aside from their operations becoming normalized, you have a massive impact of the elimination of 280E. It's a section in the tax code that treats anyone who traffics in cannabis or Schedule I substances as essentially as a drug dealer. It's a penalty. It was intended to punish drug dealers, right? It was never intended to punish state licensed cannabis operators. It predates all of that in the industry. So 280E effectively did not allow you to deduct any of your expenses as a business from your taxable income, except for your cost of goods sold. That has led to an effective tax rate of roughly 70% for cannabis operators. That's gone. That frees up a ton of cash, and there is a possibility that we're monitoring very closely that 280E may become retroactive. To me, this calls for an immediate re-rate of the industry and certainly of medical cannabis, which it applies to today. As I said, we'll see about adult use, but purely medical cannabis companies, I think their valuations should be viewed completely differently than they were the day before this order. What else is this going to do? It's just going to make life a lot easier for anybody who is a medical cannabis company. Your record keeping is simplified, albeit difficult. Your security requirements, procurement, everything. Life just got a lot easier. And most importantly, the businesses can now scale up and down the supply chain, which I I don't have a precedent for the door that just opened in any other industry overnight like this. We've covered a lot of this, right? This is limited strain diversity. That was a nice way of putting the cannabis coming out of a lot of these facilities, like any of the bulk manufacturers that are Ole Miss, is just, it hasn't been amazing. And it's been limited, extremely limited. That's gone. It's over. We will now have a normalized competitive marketplace. As far as new entrants and service providers, this just is a positive feedback loop that's going to allow everything to happen much quicker and very much at scale. Your capital goes a lot farther, right? No 280E, no unnecessary expenses. The handcuffs of Schedule 1 compliance have been completely removed. I know I only have a couple minutes here left, James, so I'm moving quickly. And I think it's really worth pointing out the accelerated timelines here. Right? Anybody who's been working in this, I'm probably preaching to the choir. It takes an extremely long time to get locations registered. We've talked about the limited supply chain, and that's all changing. It literally changed overnight. We have now a standardized registration with the DEA. We'll see, I think the jury's still out. We're going to see how fast that all happens. I am hearing anecdotally that those who have applied for DEA licenses have already been hearing back with follow-up requests from the DEA. So it looks like they are moving. The bottom line here is that this shaves years off of new drug approval pathways, right? And speeding up to, to profitability. We covered this, right? Banking, AML, all of this becomes much, much easier. Exits become realistic. IPOs become realistic. Strategic acquisitions become much more realistic. It's legal. What remains as far as risks? Smart Approaches to Marijuana has already filed a lawsuit. They hired the former Attorney General, Bill Barr, to represent them. And there will be administrative challenges. There's no question that there will be litigation challenging all of this. I will say one mitigating factor for sure is the express severability provisions, so that should any portion of the final rule get struck down, those that are not struck down will survive. As far as the timeline, June 29th, keep a close eye. I've got my popcorn ready. It begins in earnest. Hopefully it does actually conclude on July 15th. I won't be shocked if it goes a little bit longer, but what we're looking at is potentially by year end, a final rule. I'm just, I'd also note to just keep an eye on safe banking and the hemp ban as two critically important policy issues that are coming up later in the year. And then we have some actual business updates coming down the road from DeFloria, their phase 2 data. What's the investment thesis? Pretty simple. Margin expansion, huge, immediately overnight with the elimination of 280E. Research acceleration and scalability. It's a legit industry. The barriers are gone. And then eventual supply chain maturation, interstate commerce, new service providers, scaled service providers. Like I said, positive feedback loop, the halo effect may take a little bit of time, but it's here and it's happening. And of course, we have the botanical drug pathway as a complete white space for any type of drug or indication. We've only have 4 that have ever been approved, as far as I know. That I could be wrong about that, but it's minimal either way. And the optionality on adult use following the ALJ hearings. The bottom line, structural barriers to investing in this industry, scaling, and developing real medicine for people based on the cannabis plant have been greatly reduced, if not eliminated. Standard pharmaceutical frameworks. And we're in the very, very, very early, as an investor, I'd say no question, we're in the very early innings. James, we've talked about this a couple of times over the last few weeks. Like, this is really early days, and the opportunity is massive for those that the incumbents that have gotten out in front of this. And it's an exciting time. I'll leave it with that. Huge early mover advantage for certain people, but it's a huge opportunity that it's reinvigorated my love of the industry and why it reminds me why I got into the cannabis industry in the first place, because it's medicine, and it's the most exciting thing I've seen happen since I got into this business at all. So I'll leave it there. The rest of this presentation is about our firm. So if anybody wants this presentation after, I'm happy to send it, but I won't bore anybody with the Foley Hoag propaganda.
Well,
Thanks,
thank
James.
you, Jeff. We really appreciate it. The proverb, may you live in interesting times, I think applies here. I do want to note that for companies like us that are currently not doing any of what's considered psychoactive cannabinoids, molecules like THC, they're actually free currently to do that research. We are free to produce, those molecules and not worry about the scheduling that we just went through. But the scheduling is going to certainly put a big spotlight on advancing research. So thank you very much. If you want to turn your camera off, I am going to introduce Dr. Yossi Tam. Dr. Tam, while I'm introducing you, if you want to turn your camera on and get your presentation up, that would be great. Welcome. Nice to see you.
Hi, James. How are you?
Very good. Dr. Tam is at Hebrew University. He's actually presenting today from Israel. He's a full professor of pharmacology and an internationally recognized leader in metabolic disease and cannabinoid research. He holds multiple degrees. I'm not going to be able to tell you exactly what they all are, but includes masters and PhDs and a DMD degree from Hebrew University of Jerusalem. Following postdoctoral training at the National Institutes of Health, NIH, he established the Obesity and Metabolism Laboratory at the Institute for Drug Research, School of Pharmacy, Faculty of Medicine, Hebrew University in 2014. Professor Tam's pioneering work spans the endocannabinoid system, which we haven't really talked about, but that's the human body system. Endocannabinoid system's role in metabolic syndrome, organ crosstalk and metabolism, and the development of cannabinoid-based therapeutics. His exceptional productivity includes over 100 peer-reviewed publications in top-tier journals, including Nature Medicine, Cell Metabolism, Nature Communications, and JCI, among others. An H-index of 43 and 17 invented patents, 9 of which have been licensed to biotech companies, which is why we're working with Yossi, and promoted the creation of new pharmaceutical companies. Professor Tam acts as Vice President for Research, directs the Hebrew University's Multidisciplinary Center for Cannabinoid Research, and serves on scientific advisory boards for biotech and pharmaceutical companies developing next-generation metabolic therapeutics. He has received numerous national and international awards and grants, including ERC, Synergia, and NIH funding. Throughout his career, Professor Tam has integrated clinical insights with cutting-edge experimental approaches to translate basic discoveries into therapeutic strategies for obesity, diabetes, chronic kidney diseases, and fatty liver disease. As I said in my opening, there's significant research already going on. We think the door is going to open even wider, but Professor Tam is one of the people in the world that has this expertise already. And as we are thinking about who do we want to work with, and as you may be thinking about who do you want to work with, Professor Tam and his staff and his lab are primary to that. So thank you for presenting today. I will turn my camera off and leave the floor to you.
Thank you so much, James. I hope you can hear me and see my screen. Is everything okay?
Yes.
Perfect. So again, thank you for letting me present our work on obesity, and I will take these 20 minutes to tell you about why cannabinoid-based therapeutics can address obesity and the metabolic syndrome. And as you probably know, the metabolic syndrome is really a huge problem worldwide. Let's see that my presentation changes. Just a moment. Okay. Yes. It's a real problem worldwide. It involves not only obesity, but fatty liver disease, diabetes, dyslipidemia, osteoporosis, and even cancer. And basically, if we think about the metabolic syndrome, we can narrow it down to obesity and obesity epidemic, which has been a really a huge problem and nearly tripled since 1975. Almost 650 million adults around the world and almost 40 million children under the age of 5 are suffering from obesity. And you can see the projections for the United States that nearly half of the American will have obesity by 2030. So how can we manage obesity in general? The first step will be to use lifestyle modification. If this doesn't work, of course, we can think about bariatric surgery. And the last way is to utilize medication. And if we are talking about the projection that almost 50% of the US adult will become obese in less than 4 years, the global anti-obesity drug market project that anti-obesity drugs could reach almost $150 billion worldwide. And these are numbers are based on only one partial solution that is currently available, Ozempic, Wegovy, Mounjaro, the GLP-1 receptor agonist, as you may know. I would like to take this time and present you the cannabinoid system in general. So we discussed cannabinoids and cannabis. So what is the difference between them? Of course, phytocannabinoids coming from the plant, THC and CBD, and to name just the two main of phytocannabinoids. And we do have endocannabinoids, which are compounds that are produced by almost cell type in our body. And both phytocannabinoids or endocannabinoids basically modulate the cannabinoid receptors CB1 and CB2 that are present in many tissues in our body. If we try to look at the different physiological effects that are modulated by cannabis or cannabinoid, you can see that the list here is far from complete. But for today's topic, I highlighted several of these effects. And we can sure say that if we are consuming cannabis, high THC, of course, or modulating the endocannabinoid system and activate it, it makes us relax, eat, drink, rest, sleep, save and store, and conserve energy. And if we come to think about even further, all of these effects are the hallmark of what we call the thrifty phenotype. It's the same phenotype that helped humans survive evolution during frequent periods of starvation. However, in our Western society of abundant food supply, sedentary lifestyle, and increasing lifespan, these effects became the main cause in the epidemic spread of obesity, diabetes, fatty liver disease, just to name a few. So how can we utilize this information to develop cannabinoid-based medication to treat obesity, diabetes, fatty liver disease? I would like to highlight during my presentation 3 different examples of utilizing cannabis-derived product, synthetic cannabinoids, or endocannabinoid system modulator. Let's start with the first one, cannabis-derived product. And since, of course, it's not legally ethical to utilize THC because it's a psychoactive compound, let's think about other cannabinoids such as CBD, cannabidiol, or cannabigerol, CBG. Both are non-psychoactive compound present in the plant, CBD much abundant than CBG. And information about CBD and the way it regulates energy homeostasis is really quite known. However, about CBG, nobody really assesses it dramatically. I would like to show you the experimental protocol that I will utilize throughout this presentation that we are using in the lab. We take mice, put them on high-fat diet or standard diet. High-fat diet contains almost 60% of fat. And after 14 weeks of eating this diet, they become obese. The obese mice then can be treated by any type of drug that we decide. And for this experiment, we utilize CBD and CBG that are coming from the plant itself. When we tested CBD and CBG in the diet-induced obesity mouse model, we didn't see that they affect so much body weight. However, they improved their body composition by reducing fat mass and increase lean mass, muscle mass. CBD and CBG also improve glycemic control and insulin sensitivity by improving glucose tolerance, fasting blood glucose, and reduction in HOMA-IR, which is a marker of insulin resistance. CBD and CBG also improve dyslipidemia by reducing serum triglyceride, cholesterol, and the bad cholesterol, specifically LDL cholesterol. And more strikingly, CBD and CBG reduced diet-induced fatty liver disease by reducing the accumulation of fat within the liver by reducing triglycerides and as well as cholesterol, as you can see also here in these histological sections that were taken from the liver of the mice treated with CBD and CBG. So this is the first example of utilizing cannabis-derived products that have the potential to treat some of the features of the metabolic syndrome. But now I would like to feature another way of treating the metabolic syndrome by utilizing synthetic cannabinoids. I already told you that CBD has the ability to improve glycemic control, reduce dyslipidemia, as well as ameliorating fatty liver disease. But as James mentioned, CBD in the plant is not present as CBD, but as a cannabidiolic acid. The acid form, and here in red you can see the carboxylic acid that is being decarboxylized during heating to generate CBD. This molecule, CBDA, is very unstable because heating and changing in the temperature can affect its stability, and therefore there is limited pharmacological information about its effect. So we decided to protect the degradation of this compound and generated a novel synthetic cannabidiolic acid derivative that is called EPM301. We introduced a methyl group next to the amide bond to generate EPM301, which is a CBDA O-methyl ester. It's a novel CBDA analog, which has the ability to increase the stability of CBDA. It has a superior oral bioavailability compared to CBD, and when it was tested in models for anxiety and depression, it shows that it also rescued these behavioral effects. So we decided to test EPM301 in our diet-induced obesity, and to make a story short, we found that it's very efficacious. However, can CBDA, O-methyl ester, or EPM301 can treat genetic-induced obesity? And here I would like to introduce you a new disease, or a rare disease called Prader-Willi syndrome, PWS, that is a rare genetic disorder that is caused by the loss of paternally inherited cluster of genes on chromosome 15. Patients that suffer from PWS have shown profound hyperphagia and morbid obesity, and therefore it makes sense that we would like to treat their syndrome utilizing any type of drug, and in this case, CBDA O-methyl ester. In a prior study, we measured endocannabinoids in the circulation of patients suffering from PWS, and we found that 2-AG, the main endocannabinoid, is significantly increased in PWS patients compared to normal obese patients that were linked to their hyperphagia and obesity. So we decided to test EPM301 in a mouse model for PWS. We gave daily administration of this drug for almost 15 weeks, and we found that EPM301, the synthetic cannabidiolic acid derivative, is able to reduce body weight gain. It reduces fat mass and increases lean muscle mass. So this was the second example of cannabinoid-based medication, and now let's move to endocannabinoid system modulator. I already showed you that the effects that are modulated by cannabinoids are basically modulated through the CB1 receptor. CB1 receptor is the main receptor that is present in the human brain, but also in periphery. And therefore, if CB1 activation causes us to relax, eat, drink, rest, sleep, save and store and conserve energy, why not trying to block it? And in fact, one such drug that blocks the CB1 receptor was developed already by Sanofi. A drug called rimonabant or Acomplia is a specific CB1 receptor blocker that blocks this receptor and was marketed to treat human obesity and diabetes. Rimonabant was considered a wonder drug that can lose your body weight and maintain it for a long period of time. However, patients that have been taking rimonabant also reported anxiety, depression, and suicidal ideation. And think about it, if you activate CB1 through THC and it makes you basically to get stoned and happy, if you block the receptor, it will induce anxiety and depression. And therefore, the FDA rejected the drug to treat obesity, although it was the first blockbuster before GLP-1 receptor agonist was in the market. And not only rimonabant withdrew from the market, but also taranabant, otenabant, ibipinabant, all the rimonabant-like compounds that try to block CB1 receptor in the brain. But as I told you, CB1 is not only present in the brain, it's also present in many peripheral organs. But we knew 10, 15 years ago that only CB1 in the brain modulates food intake and motivation to eat, to reduce energy expenditure, with no information about the role of CB1 receptor in peripheral organs. So to make a story short, throughout extensive work that my lab has done and many other labs in the world revealed that basically CB1 receptor in peripheral organs, in the gut, in the liver, bone, adipose tissue, kidney, and muscle is important for the development also of different features of the metabolic syndrome. And this has basically raised the possibility to target the CB1 receptor in peripheral organs without penetrating the brain. And since I'm a pharmacologist by training, I always like to cite Sir James Blake, who won the Nobel Prize in 1988, who said that the most fruitful basis for the discovery of a new drug is to start with an old drug. And this is exactly what we did. We took the core structure of every CB1 blocker that was developed by other pharma company, and we decided to change it chemically in order to reduce the blood-brain barrier penetration, to retain the high CB1 affinity, and to maintain the essential druggable properties. We started our screening with thousands of different compounds. We screened them based on high CB1 affinity and selectivity, low blood-brain barrier penetration in vivo, appropriate tissue distribution, in vivo efficacy studies, until we found lead molecules which are both efficacious and safe. I would like to introduce you one such compound called BNS-822. It's a peripherally restricted CB1 antagonist, and when it was given to diet-induced obese mice, it dramatically 20 and 25% of weight loss, reduce of food intake, fat mass is reduced while lean mass is basically increased. BNS-822 also was able to prevent fatty liver disease by reducing hepatic triglyceride and accumulation of fat within the liver, and BNS-822 was able to improve metabolic profile in obese mice by reducing circulating triglyceride, LDL, cholesterol, and improving the ratio between the good and bad cholesterol. All of these effects were associated with basically no penetration of BNS-822 to the brain, as you can see it here, comparison to rimonabant, and without the induction of brain-mediated side effects like rimonabant does. It induces ambulation in mice. BNS-822 is really unique in this feature because it does not penetrate the brain. It does not induce CNS-mediated side effects. In fact, utilizing peripheral CB1 receptor antagonists, we were able to treat almost the entire metabolic syndrome, starting from obesity, fatty liver disease, diabetes, and so on throughout hundreds of different publications. So to conclude my presentation, I would like to highlight the cannabinoid-based therapeutics to treat the metabolic syndrome. As you know, to generate novel drug, it starts with pre-discovery idea, that after that, a stage of drug discovery, preclinical stage start until IND is submitted to the FDA, and then clinical trials are being initiated in humans until NDA is being submitted to the FDA. Would like to highlight that our work with CBD and CBG coming from the plant are now at the final stage of preclinical assessment, and they will be developed further to the clinic through Cummins Biopharma, a US-based company. The CBDA O-methyl ester, or EPM 301 drug, has now received a orphan drug designation by the FDA and rare pediatric disease designation by the EMA, the European Agency, to initiate clinical trials in PWS patients. And this is being developed through EPM Therapeutics, and the peripheral CB1 receptor blockers are also entering phase 1 clinical trial via BioNanoSim and CurisAI, two Israeli-based companies that will take it into the market. So with that, I would like to conclude my presentation and, of course, acknowledge my mentors, my current lab members, the amazing team at the Hebrew University, former lab members, the funding agencies, the collaborators, and the companies that I believe will bring, at the end, cannabinoid-based therapeutics to the market to treat human obesity and metabolic syndrome. Thank you so much, and I would be happy to answer any questions that you may have. James, I cannot hear you.
I bet that's better now.
Yes.
Thank you very much for your presentation. We really appreciate your time and giving everybody your knowledge about endocannabinoid system and how to apply cannabinoids and cannabinoid medicine. Professor Tam mentioned, that we obviously have a relationship and we want to provide material for future studies, obviously with the funding that they're looking for and we're trying to help with, that can be achieved. You mentioned a couple of commercial companies. You will hear some presented today. CitraChem has memorandums of understanding with to be a provider. One of those is Carmen's that was just mentioned as licensing one of Hebrew University's compounds. So, we have an integrated relationship here, and this is evidence of how we CitraChem want to be a good provider. Maybe you could just tell the audience a little bit, why it's important to have the kind of material that we can provide for your research.
I think, first of all, the ability to have compounds that are pure and they are validated and with large quantities, this is an ability that we can basically conduct research. And if we want to move further to the clinic, we will need, of course, hundreds, if not kilograms of those compounds to run clinical trials. And we need a validated company that will be able to provide this. CBD and CBG that can be generated from the plant, of course, it will take a lot of time and efforts to generate large quantities of these compounds just by extracting them from the plant. We can see the hurdles that GW or Jazz Pharmaceuticals are basically facing with Epidiolex to generate only one compound, the CBD, to treat Dravet syndrome and Lennox-Gastaut. So if we want to treat and utilizing this compound, for example, for the obesity market, which is a much larger population that we need to treat, we will need larger quantities that will not be able to achieve by just growing the plant.
Right. That is exactly correct for us. So we appreciate that you recognize that. I appreciate your time, and we're going to move to Jason Kingsbury presenting here in the office. So let me switch to our screen sharing mode. Give me 1 second. Okay. We are definitely sharing, and you want to see this. All right, I believe you can see the agenda. And I want to, I can't get to it. Pardon me while I wait for technology. There we go. So let me introduce Dr. Jason Kingsbury. He is CitraChem's co-founder, principal investigator, and chief scientific officer. His initial invention, which became CitraChem's platform, is focused on flavonoid and terpene natural products as medicinal agents. Jason was a fellow NSF and government assistance in areas of national need, a fellow during doctoral work at Boston College with Amir Hoveda. His thesis work culminated in work making him an expert in chiral ligand design and asymmetric catalysts, each subjects of vital importance in CitraChem's processes. Dr. Kingsbury's NIH-supported postdoctoral work at Harvard involved total synthesis of 2 members of a family of bioactive natural products. I'll just keep emphasizing he's an expert in natural products. Starting his independent career in 2006, Jason's laboratory utilizes BC startup funds and a grant from the ACS Petroleum Research Fund. In 2016, he was a lead technical advisor on efforts to scale up a library of biocompatible DNA and RNA binding fluorophores in collaboration with scientists at the University of Washington. In 2019, he tested a reaction which led to a novel intermediate to facilitate production of minor phytocannabinoids and is now one of the unique claims in CitraChem's patent applications. So here in Norwood headquarters is Dr. Jason Kingsbury.
Great. Good morning. Just want to sincerely thank everyone for coming together in this virtual format on short notice. And congratulations to Yossi and his team for continuing to invigorate a very long and rich history of cannabis-based medicine at Hebrew University. I'm Jason, really looking forward for the chance to get to know some of you hearing about CitraChem for the first time. And as its chief science officer, I'm excited to temporarily switch gears and transition our discussion in the direction of commercialization of these molecules that you're hearing about. As James noted in the introduction, I remain a very motivated and research active synthetic chemist, and I feel fortunate to have trained under some of the older legends in the field. As such, I feel that some of the ideals and beliefs that I may subconsciously carry have been important in how we've gone about branding CitraChem as a team. And one of those is demonstrated in this quote here. So without a doubt, purity is a matter of degree. This is not a new concept to anyone close to the life sciences, but it also happens to be the opening quote in what many practitioners of synthesis consider the Bible, the go-to handbook on how to purify any organic substance. So in coining this quote, the authors were essentially cautioning us. They want to remind us that absolute perfection or 100% purity may be unattainable. When you hear and see claims that approach this ideal, it's important to realize that there are inherent sources of experimental error. For example, making mass measurements at a balance. And these experimental errors factor into these calculations around purity. Another limitation is that we always must be working within the limits of detection with very sensitive instrumentation. And this is very important to us because, as you'll see, we've deliberately chosen to target and scale up these phytocannabinoids and derivatives completely outside of the plant. A second important conviction of ours is that a molecule is a molecule. Whether you like to drink coffee or choose to open a Red Bull before you work out, we're obviously bringing in to our system caffeine as a stimulant. And so one of the things that remains curious to me is how there are fierce proponents of the plant as somehow having magical or supernatural powers. And I think one of the explanations for that is part of our history. It turns out that in the mid-1800s, there was a concept known as vitalism. And this was a medical belief that only living organisms that contained some special power or living force were capable of synthesizing organic compounds. And this theory was refuted the moment serendipity struck. Frederick Wohler, the University of Göttingen, was experimenting with a lead cyanate salt that he happened to expose to ammonia and water. And after some detective work, he found that a metathesis, an adjoining of the cyanate anion with an ammonium counterion from ammonia, gave this salt that underwent isomerization into urea. So isomers are structures that have the same molecular formula, but are somehow different in terms of their connectivity or structure. So the moment that Wohler discovered that something widely associated with living organisms only, or organic, could be produced in a test tube or flask outside of a living organism, was the decline of vitalism, but importantly, it also marked the birth of the field of organic synthesis as we know it today. One of the things I'd like to do is cycle back now and to that tenet around approaching high purity, which is critically important in human medicine. As Jeff shared in talk number 2, for many years, the US industry has been regulated in a way that determines how the plant itself is farmed and manipulated. And one of the challenges is that in the face of increasing climate change, increasing scarcity of precious natural resources like land, water, and energy, agriculture is becoming an even more variable practice. There are issues associated with the use of pesticides, possibility for contamination by mold and other pathogens, and even the potential for plants to take up heavy metals from the soil. So we believe that a broadly applicable and highly scalable manufacturing platform will surely win the day and deliver molecules without any unknown contaminants. One of the reasons for this, as you'll see, is that we know precisely what our raw material inputs are. We specifically choose chemical reagents that are introduced to the batch process from a given commercial supplier that we can establish a reliable supply chain with. And most importantly, these chemical transformations that I'll begin to show you are well understood in terms of how they impact or affect chemical change. This is something that we call mechanism. And so one of the things that James asked me to do is really present a backdrop, an origin story, if you will, on really how and why we set out to develop such a manufacturing platform. Shown here on this next slide, sorry, let's go back one. So on this slide, this obviously needs no introduction, but The message is that this area of clinical medicine has already recorded major successes, of which I show only two here. Marinol was first developed by AbbVie. This is a synthetic, fully synthetic THC. Goes by the generic name dronabinol. And it was FDA approved in the mid-1980s for use as an antiemetic for cancer patients undergoing chemotherapy. Shortly thereafter, another FDA-approved indication followed, and this was for appetite stimulation in AIDS patients. This morning, we've already heard a lot about GW Pharma's development of a botanically sourced oral solution of CBD, which is important in treating refractory or intractable forms of youth epilepsy. And I think I just want to add that subsequent to the acquisition by Jazz, it's important to realize that GW Pharma had a method of use patent, and within that patent, there was clear provisions set out for, in the future, allowing a fully synthetic CBD to be part of this medicinal formulation. Yossi also has already pointed out that, and in the words of Jeff, this is just the tip of the iceberg. What you'll notice is that these are the neutral major molecules that have simply been the subject of focus for researchers for more years. And in fact, the plant is a veritable chemical factory of the so-called cannabinoid acids. Shown down here is a schematic that just brings up the identity of all these acronyms that terminate in A. So that A is referring to the presence of this carboxyl or acid function, which is prone to being lost in the form of carbon dioxide in a process called decarboxylation. This is accelerated if we have plant material or isolates, extracts of the plant that have been overly processed, subject to prolonged storage, or exposed, however inadvertently, to heat and sunlight. And so I want to take you all back to about 2019, just before the COVID pandemic. I was and in discussions with my co-founders, I shared my vision, and that was to target these molecules in a way that would rely on the typical blue terpene and green aromatic fragments. And we set out, just as it is in nature, to merge or unite these two fragments more or less at the 50-yard line. And in a way, we were also motivated to retain that carboxyl group or look at suitable derivatives that would enhance its stability using a similar strategy to the one that has been furthered to licensed compounds at Hebrew University. This became our roadmap, if you will, for proof of concept. Choosing to begin with citral, which is a known fragrance molecule. It's widely abundant and it's very inexpensive. We set out to discover a reagent that would cyclize this small molecule raw material and form the hexagonal ring that you see pictured in this electron deficient intermediate in green. The reason that I was aware of this intermediate and building block is that more or less all of the primary and patent literature had enabled access to this intermediate using what we're going to call PMD for short. You take this other terpenol and you expose it to a desiccant or a dehydrating agent, you lose formally the equivalent of water, and you arrive at this stereo-defined intermediate, which is amenable to coupling using a well-known reaction. So our priorities were to move away from PMD, uncover a new or a novel pathway for accessing this intermediate. And along the way, I set out to also modify some of the known protocols for building these aromatic resorcinol compounds in a way that would retain that carboxyl ester or acid group. Fast forward a few months, and with the support of angels, we were able to file an international provisional patent application that's now undergoing examination in these various jurisdictions shown here. You can imagine that at the time, I was really excited and becoming very well read about how to access PMD and whether there would be any opportunities for streamlining its preparation. Shown on this slide are the two broad major categories of synthesis. And the first is something that we call a linear synthesis. For those that are inclined, I show the elaboration in the structure of the raw material through three intermediates arriving at this final building block shown here. And you don't need to know too much chemistry to actually connect with an important mathematical lesson here. One of the challenges with a linear synthesis is that there is a dramatic diminishing in your overall yield as you add additional steps to this process. So it turns out that for a linear sequence, your overall yield is a function of your individual yield for each step, basically times one another. So if you take the fraction 0.80 and you put it to the fourth power, 0.8 times 0.8 times 0.8 times 0.8 again gives 0.41. And you're left wondering, where did over half of my theoretical return suddenly vanish to? The situation is even worse if the yield per step still is dropping, but considered what many might believe is an acceptable yield or a publishable yield in the literature. So with a 55% yield over those four steps, you're now in the single digits in terms of your overall or net yield. So one of the ways that you combat linearity, and we learned this very early in graduate school, is to build in what we call convergency. This is where now you're taking two or more building blocks and bringing them together in a combination that's shown or schematically represented here. Notice that with respect to chemical entities, there were 5 blocks or building blocks shown here. We've now switched, and A and C are both raw material inputs, B and D are intermediates, and the final output is again E. But importantly, there's only 3 discrete operations or steps. And it turns out that your yield overall is going to be a function of only 2 of those steps. One of these, either A to B or C to D, will be higher yielding and won't factor into your net yield. Take a look at the jump in efficiency. So if we keep the same respectable or good 80% yield over 2 steps, now we're getting 64% overall, which is far and away higher than the 41%, almost 1.5 times. And even if we drop to that 55% yield over 2 steps, we're still getting more than triple the amount of material in a linear sequence. So again, these are just representations. And on the bottom, I've shown how our process is a 2-step approach to what is an allylic alcohol isomer of the well-known PMD. I'll show you what this looks like in just a moment. I also want to just point out that This is widely viewed as the state of the art. These building blocks shown here on the route to CBD are part of a Noramco patent from 2018. Noramco now has a subsidiary known as Purasis. And even beyond the linear approach to PMD, what Purasis chose to do to target cannabidiol is install blocking groups to ensure that bond formation was at the right position. And that also requires removing those bromine groups later. And so our template for success is going to be using our own approach to this monoterpenol, access the same intermediate that one obtains from PMD on dehydration, and again, retain the acid group so we can access cannabinoid acids like CBDA, which in turn is a 2-for-1, right? If we have a highly efficient approach to CBDA, we can use this thermal decarboxylation to get CBD as well. Very briefly, I'm going to summarize our capabilities here in bullet format. We sought out and are now partnered with a US-based full-service CDMO, development and manufacturing organization. And in a matter of months, we've been able to bring 3 cannabinoid APIs into a scale-up mode. This was initially done under non-GMP conditions, but we have kilogram quantities of each of these cannabinoids shown over here. We are pharma-focused, and as I just mentioned, our routes feature half as many steps as our competitors. I'd really like to thank Wendy Young and Alex Breton, who we met at some conferences here in Boston over the last couple of years. This is a female-led analytical lab in Colorado who's done all of our COA work, and they are really priming us to be ready to go for this elaborate standardization process. So we know exactly if there are any trace impurities, what they are, and how to lower those detection limits or eradicate any byproducts completely. Where we plan to go with this is ultimately ensure that our API costs in final form are competitive with botanical isolates. And as you're hearing, we'd like to target rare and minor cannabinoids that are of low natural abundance in the plant and continue to partner with other early stage drug discovery companies to bolster intellectual property and really exploit a lot of the R&D intelligence that we're gaining through our MedChem work. So I'm going to close with a visual perspective of our platform and how we're hoping to basically rinse, wash, repeat, right? Change inputs, expand on the diversity of molecules that we can make, and begin targeting new compositions of matter. My dear friend and close colleague, Mark Scialdone, is on deck, and Mark is super passionate about novel compositions of matter and exploring some of the unknown potentials that they unlock. So again, we really view citral as a raw material that forms the basis for everything we do. Going up on this slide, we have a one-step approach from this reduced form of citral and another commercial compound known as olivitol, and we can achieve a 70% yield of CBG with these potential clinical applications. I'm then going to bring your attention down and show that from citral, there's another one-step process that we've developed by modifying what's known in the literature to make CBC. CBC is really exciting because it has this chromine scaffold. Chromines are what we might call privileged natural product scaffolds that are of very highly useful and variable biological activity. Finally, as we move left to right in this screen, we can start to approach what we're going to call the majors, right? Those molecules that have a cyclized and stereochemically important cyclohexene ring. So the blueprint of what we've actually accomplished at the mechanistic level is shown here. I was ultimately able to identify that aluminum-3 based Lewis acid systems can accomplish a paracyclic intramolecular reaction shown here. And we also were able to capitalize on an accident. It turns out that we are able to adjust the oxidation state of the major product, which is the ketone, and through an asymmetric reduction, establish this trans or opposite orientation of the groups attached to the lower right portion of this intermediate we call iso. Finally, either resorcinolic esters or acids are our next input. We work hard to eliminate the use of any blocking or protecting groups. So in one step, we're able to access CBDA with heat providing the decarboxylation necessary to afford CBD. And just as a point of interest, our approach, which was part of our original patent filing, was also to target the methyl ester of CBDA. We fully understand and celebrate that this is a Hebrew University composition, I think advanced by Raphael Meshalam, and has now been licensed to EPM Therapeutics for this potential treatment of hyperphagia and Prader-Willi syndrome. With that, I'll just close by saying we are very excited to constantly have MedChem and R&D operations going on just behind the door here. We have a roughly 1,000 square foot lab outfitted with fume hoods, inert gas setups, and all the reagents that are currently supporting these partnerships. I also just want to thank all of our collaborators for coming together in this format. Be happy to answer any questions. Thank you.
Thank you, Jason. I don't believe any questions online. I have lots of questions, but we can talk later.
Okay, sounds good.
Thank you very much. I'm going to stop sharing. And then I will do an intro for Mark Sheldon, who will get his presentation going. Our technology doesn't like to have me stop sharing. Okay, so Mark, while you are teeing up your presentation, I'm going to do a nice introduction for you. How are you? Mark is up in Colchester, Vermont.
Can you hear me?
We can hear you.
Okay, awesome.
So Dr. Sheldon is an organic chemist with extensive industrial research experience in solving complex problems and driving programs to successful conclusions. He is highly skilled at conceiving of, designing, synthesizing, and characterizing a wide variety of new molecules in product formulations. He is highly adept at reaction troubleshooting, optimization, and process scale-up to several hundred kilogram scale. Known for the generation of prototype efficacy and safety assessment testing results essential to product commercialization. Mark is known for his excellent industrial chemistry insight, focus, communication, leadership, and team-building skills. He has demonstrated success in developing valuable intellectual property, obtaining product registrations and certifications, initiating and sustaining customer engagement during product launch, and delivering technical service. Mark is currently a research scientist in residence at St. Michael's College in Colchester, Vermont, which includes conducting contracted research for CitraChem. Mark enjoyed an 18-year career at DuPont as a research chemist. He earned his bachelor's and master's degrees in chemistry at Michigan Technological University, his PhD from Wayne State University, and was a postdoc at Colorado State University. Mark, if you want to put it in presenter mode, that's great. Otherwise, we can see your slide as is. And welcome to the session. You are on mute if you want to unmute.
Okay.
There you go.
You can hear
Floor
me
is
now?
yours.
All right, and I'll put it in slideshow.
Great.
Okay.
Perfect.
Can you see it?
Yes, sir.
Okay. All right. Well, good morning, everyone, or good afternoon, depending on where you're at. My name is Mark Sheldon, and yeah, as James said, I'm a research scientist in residence here at St. Michael's College. St. Michael's College is a small little 4-year liberal arts college in Colchester, Vermont. And I'm sitting here in Churay Science Hall, which is basically where we have both chemistry and biology, as well as physics too, as well. Okay, so today I'm going to tell you about a project that we're looking at for the treatment of traumatic brain injury using the intranasal delivery of cannabinoids. So, intranasal administration, we believe, is the delivery of choice to get cannabinoids into the brain rapidly after a traumatic brain injury. That could be something like a concussion on a football field, but it could also be something that soldiers would experience in the theater, right? So anytime traumatic brain injury happens, there's what's called the golden hour of trauma, which is kind of like that hour that's just after traumatic brain injury happens. And so what we hypothesize is that if we can get cannabinoids into the brain during that golden hour of trauma, that the cannabinoids are known to reduce neuroinflammation, and this reduction in neuroinflammation and brain swelling, we believe, will create a better outcome for the patient. So, our particular focus is to use nanoemulsions. Nanoemulsion, when you think about nanoemulsion, think of just dairy milk, right? It's emulsified oil in a water phase, and so something like milk is mostly water, right? But the oil that's in there has basically been dispersed through the surfactant proteins that exist in dairy milk, right? So you're probably aware or have seen the increase in popularity in cannabinoid-based beverages, particularly in states like Minnesota, where now you can get beverages at cannabinoid-based beverages at liquor stores and at places that usually handles things like beer, wine, and spirits, as well as establishments too. So you can actually go to an establishment like a bar where you would traditionally get alcohol drinks and now get cannabis-based drinks. Well, what's made that beverage market sector really take off is this development of nanoemulsions that are stable. You know, cannabinoids aren't very soluble in water, so they have to be emulsified. And the nanoemulsions really add two very critical properties. They increase the overall bioavailability. This is something that I think we touched on previously when we were thinking about CBD and CBDA. I think there's data to show that the cannabinoid acids have an enhanced bioavailability, probably due to the fact that CBD is rather insoluble in water, but when you add that acid group, now you have something that can ionize at physiological pH and will help with bioavailability. Well, the nanoemulsions take it to the next level because the nanoemulsion is basically a formulation vehicle that creates these emulsified formulations that have very, very small particle size. So they're able to, traverse the cell wall and reveal their contents on the other side. So I think that increase in bioavailability coupled with a rapid onset of action, this is also something that, again, is being manifested in this cannabis drink market sector because, you can't usually, if you take an edible, it's going to take some time before it gets through your digestive system, gets through the liver and works its way into the bloodstream, and then it's got to get to the brain in order to feel psychoactivity. What I can tell you is that these nanoemulsions increase the rate at which a consumer can feel the psychoactivity. So halfway through that beverage, even on like a 10 milligram beverage, you're going to start to feel the psychoactivity of a nano-emulsified cannabinoid. Okay, the other thing I just wanted to mention too is that, we wanted to also think about the cannabinoids, as Professor Tam pointed out, the cannabinoid acids are also enjoy a different set of pharmacology than the decarboxylated cannabinoids, and they both act to basically reduce inflammation. So we would want to maybe consider using both decarboxylated as well as carboxylated cannabinoids in this approach. Getting a carboxylated cannabinoid into the brain has been somewhat of a challenge because that carboxylic acid group creates a polar functional group that typically does not lend itself to traversing the blood-brain barrier. When you consume cannabis, the heat from the fire, from the lighter or the matches that you're using to smoke that joint or bowl or whatever, is heating up the cannabinoids to basically decarboxylate them. So you're not getting cannabinoid acids across the blood-brain barrier. Perhaps doing it in a nanoemulsion can be a suitable way of doing that. Okay, so a couple of years back, we partnered with a group in Minnesota called Health Partners. They used to be part of the University of Minnesota, but they've sort of split off, and they're a nonprofit that does a lot of preclinical and clinical testing for the biopharmaceutical, pharmaceutical, and medical device industry. Collaborated with this group in their neuroscience research institute that are world-renowned experts for intranasal administration, looking at mostly neurodegenerative diseases. These are the folks who developed the transnasal delivery of insulin to treat Alzheimer's disease. So they approached us. They were interested in using intranasal administration to deliver cannabinoids. At the time in 2020, this was after the Farm Bill was passed, but before the recent change in scheduling. So for regulatory reasons and for legal reasons, we decided to stick to cannabidiol, but they were particularly interested in some work that we had done on cannabinoid hydrogenation. So I'll talk a little bit more about that in just a little bit, but hydrogenation is a process that you can apply to CBD to convert CBD into what we call tetrahydrocannabidiol, or THCBD, right? And so we took both of these molecules, put a radioactive label on them so we could measure the amount that's administered to the brain after intranasal delivery. And what you're looking at here is a graph of concentration of the CBD and tetrahydro CBD that were in these various sections of the brain. So here you can see the olfactory bulbs. Here's the cortex, here's mid-cortex. You can see the hippocampus here, septum here. And what you can see is that we're delivering not just the CBD, but also the tetrahydro CBD in concentrations above micromolar levels, where we know that these types of cannabinoids have activity at the cannabinoid receptors. So if this were THC that we had delivered here, we would be delivering THC above its binding constant to the CB1 receptor. These are also indicative of using intranasal delivery for, in this case, this is the liver, here's the kidney, and here's the spleen, the lung, and the blood of the mice. And what you could see is through intranasal delivery that we are successful at getting micromolar concentrations of these cannabinoids into not just the brain, but these organs as well. So we're currently teeing up a next step, being a feasibility dose finding study where we're going to use now that previous formulation was just nothing more than olive oil in water. But now we're going to basically use this nanoemulsion idea to nanoemulsify the cannabinoids in what's called a closed head TBI model in rats. This will be done with our friends at Health Partners in Minnesota. We're currently writing an STTR grant proposal looking at a fall 2026 submission. Ultimately, we'd like to apply this intranasal delivery to other brain disorders that would include things like stroke, glioblastoma multiforme, neurodegenerative disease such as Parkinson's and Alzheimer's, and other forms of dementia. We'd like to develop this using the cannabinoids that we've talked about, like the CBD and perhaps CBDA would be a very interesting combination to have both CBD and CBDA in a nanoemulsion for the treatment of traumatic brain injury. But we're also interested in developing new proprietary derivatives, so new derivatives of phytocannabinoids. Right now, we're active in cancer screening, and we're basically demonstrating the hypothesis that the chemical modification of these compounds improves their activity. So we've been primarily focused on the double bond, which is prone to isomerization, right? We've heard a little bit about delta 9 and delta 8. And so what we're interested in doing is stabilizing this functional group here. We've done so by looking at chemical modification by hydrogenation of that double bond, and we've now extended this to hydrofluorination of the double bond, where it puts a fluorine group on this tertiary position. We've also looked at some chemistries looking at the esterification, and we've talked a little bit before about the methyl ester of CBDA. We've made some methyl ester or just simple alkyl esters. We've also extended it to amides too as well, so we could make amide groups of this carboxylate too as well. So here are some of the new derivatives. Most notably, we are the inventors and we hold the composition of matter patent on hexahydrocannabinolic acid. I don't know if you could see my screen, but this big jar right here is hexahydrocannabinolic acid, right? Nice fluffy white crystalline solid. We get this from the hydrogenation of THCA. When we do that, we generate two stereoisomers, the 9R and the 9S. And in collaboration with my colleague Lindsay Avery here at St. Michael's, we've been looking at the differential activity, and it's very interesting because with HHC, it's known that the R isomer is more active at CB1 than the S isomer. But with HHCA, what we're finding out is that the S isomer is a better killer of cancer cells and Jurkat cells than the R isomer. So we're currently writing that up for publication. We've recently, again, extended this functionalization of the double bond to hydrofluorination. So when you hydrofluorinate THCA, you make this 9-fluoro-HHCA. So just HHCA now with just a fluorine atom here. And what's interesting about fluorine is, fluorine has long been used as an isoster for hydrogen by medicinal chemists. It's a very different atom in that it lives at the northeast corner of the periodic table, so it's the most electronegative element on the periodic table, and its electronegativity certainly affects the electronic parameters of the rings that it's attached to. So when you take 9-FHHCA and you thermally decarboxylate it, it makes 9-FHHC. So this is just HHC with a fluorine atom at the 9 position. We could make a similar compound with the Varin series with a 3-carbon tail. This is 9-FHHCV. And this is a monofluoro derivative of CBC where we've fluorinated the 9 position here. Actually, this is derived from the hydrofluorination of CBG. But what happens in that reaction is we do hydrofluorinate the position over here, the 9 position, but it snaps shut to give this benzopyrene, and we lose that fluorine. We're able to keep one fluorine on the molecule, but we're able to basically make this type of pyrene structure. So these compounds, we've sent these now to the NCI 60. This is the National Cancer Institute Developmental Therapeutics Program that will give you cancer data. And what we see here is that the 9-fluoro-HHC is a very broad, potent anti-cancer drug. So it's basically showing, when you see the full bars like this here, it's killing all of these cancer cells, all of these cancer lines. So it's a pretty good broad spectrum killer. Here's the 9-FHHCV, so you can see it's not as broad spectrum, but still potent killers of. In this case, this is leukemia. I believe these are. Let's see. These are non-small cell lung cancers. This is colon cancer here. This is brain cancer, CNS cancer. This is melanoma, ovarian cancer, renal cancer, prostate cancer, and breast cancer down here colored in pink. So we're beginning to look at how these compounds might have selectivity. In this case, what we're seeing is that the IC50 of our most active compounds, in this case, the 9-FHHC against this leukemia cell line, has got a 131 nanomolar IC50, and we see about twice that with the 243 nanomolar IC50 for 9-FHHCV. So we're seeing some selectivity between the different lines of leukemia, which is interesting. When you see the bar go down like this means that we're killing it. In this case, we're killing it here. We're maybe not as good here. So again, maybe a little selectivity between 9-FHHCV and 9-FHHC. So we've just begun to look at, this is with a collaborator in Colorado who's doing some simulated docking studies for us in the CB1 and CB2 receptor, so we can do some rational drug design arguments on why we might make certain compounds versus another to enhance activity. So that's my presentation today. I'm happy to answer any questions that you may have.
Appreciate that, Mark. I do not see questions in the queue. I just want to emphasize that, Mark's a great partner both in doing some development work on molecules that CitraChem's interested in, and then working with us on how CitraChem can participate in helping make the molecules you just saw manufacturable at a commercial level. So we have a symbiotic relationship going with Mark and St. Michael's. Appreciate that. Enjoy Vermont.
Thank you.
Okay, our next presenter is Jeff Heron. Sorry. Jeff, if you are online and want to start your presentation material, I will do your introduction. I am looking for you. Not see. Oh, there you are. Look, it says James Winokur. That's great. It's a great name. Jeff is the CEO of Greenway Herbal Products, which is the operator of the Tennessee University developed third-party tested CBDA and CBD 1-to-1 formula. We talked about that earlier. but Jeff's going to go into detail. Backed by Middle Tennessee State University Center for Botanical Medicine Research, their patent-protected innovations bridge rigorous science and real-world results. Jeff is an expert in cross-sector commercialization from IP through licensing, e-commerce, and traditional channels. Previously, he held roles fostering innovation at Ananda Scientific Neurotherapeutics, also known as Oryx Wound Care Technology, and Vandemont, related to their medical cannabis business strategy. Jeff is a champion for science-first solutions that empower healthier living. He earned his finance degree from Belmont University in Nashville. Jeff, I know, is visiting New England, so we appreciate you taking time out to do this presentation. But I think the audience is going to be excited to see there's a patented formulation going through the FDA path. So if you want to get your slides up, I will turn my camera off.
Can you see my slides now?
We cannot yet see your slides.
Can you pull them up?
I can. Would you like me to share and then you can just ask me to advance?
If you can, that'd be great. Just because of the conflict with link I used, I obviously jumped in under your link of why there's the name conflict. So there we go. I can see it.
Okay, just let
Can
me
you
know
hear
when you'd
me
like me to
okay?
advance. Yes, sir.
All right. I will just say next slide when we're ready. So first, I would like to say thank you to the CitraChem team for organizing this and for the hard work that Jason has done in developing this to a scalable solution, that the path that you guys are on. I know that these molecules are plentiful in the origination source naturally, and then mimicking them and bringing them to market the way in which you guys have proposed is very exciting. So basically what you've heard is the chemistry this morning, and what I'm here to show you guys on this presentation today is what these molecules will build towards. The patented pharmaceutical asset that we are moving down with an FDA development path is what we are actively doing today with a regulatory agency, and FDA, ex-FDA, and ex-CEDAR Council. We are active in that today, working on gap analysis for determining the exact indication that we're going to focus on. The next slide, please. What if the most important word in pharmaceutical development right now isn't just a molecule, it's a ratio? The synergy that is identified in the ratio of this methods of use patent that we hold is one that is very unique in that the synergy was identified in combination, and it allowed us to go down a further discovery pathway and really focus in on the acidic precursor form of cannabidiol, which is cannabidiolic acid. When combined at a precise 1-to-1 equal molar ratio, you definitely get something that neither one does individually. And that is what spurred the university relationship to file the patent application initially, and then later assigned it to our company because of many factors, primarily that we wanted to pursue international filings, which is very unique in that they are issuing internationally even as we speak. Next slide, please. So the 1-to-1 cannabidiol and cannabidiolic acid produces a cytokine suppression efficacy much greater than CBD alone. And it is approximately 2x cytokine suppression from our in vivo models, murine models, And that synergy is really what drives the identification of what indication we're going to focus on. The one-to-one ratio is a mechanism, and if you do remove it within a ratio, there is a buffer there within the ratio, but you do lose the effect or that synergy. And so we've pushed the boundaries in that to identify where that is, and it's all relative and present in our patent application. The cytokine dysregulation is shared across autoimmune disease, and we focused on the inflammation that autoimmune diseases present, and the patent claims initially issued are autoimmune disease inflammation focused. Next slide, please. So it's a defined spectrum. There's in the cannabis world, there's what is called full spectrum. There's broad spectrum, which is essentially a step down by the removal of the tetrahydrocannabidiol. And then there's this new terminology that we're using called a defined spectrum. It is a very precise equimolar ratio of cannabinoids, and being able to identify a defined spectrum allows us to really focus on that synergy as the deliverable that one would receive through consumption. So precisely characterized, it is the reproducibility of the synthesized mechanism and the pharma-grade defined spectrum that CitraChem affords us that we are excited about the technology that CitraChem has. Next slide, please. So in 2023, the methods of use patent issued in the United Kingdom for the autoimmune disease, anti-inflammatory, with all the autoimmune diseases. There's about 16 or so that are named within the patent. The United States patent issued in December of 2025, and then the European Union patents issued in April of this year. So we're very active and very early stage in our FDA drug development pathway. We are really focused on the FDA pathway today as it relates to now confidently moving forward with the methods of use patent, and we are active in the prosecution of more claims. These are ongoing activities today that, from a business side, not a science side, we are actively defending all of the 84 claims in the original patent application. Next slide, please. So truly, the science that we have been able to identify is only as strong as the chemistry behind it. Without the purity in the molecules and the combination in that purity, our intellectual property does not hold. And therefore, what CitraChem brings to the table is its formulation capabilities that can strengthen our intellectual property with confidence, even through scale and through the delivery mechanisms that are plentiful, as there are different ways to consume these molecules. And we are looking at oral in pill form, tablet form. We're looking at soft gel form. We have explored the nano-emulsion water-soluble form. We've experimented with all forms, and we are currently narrowing that focus as we enter the IND engagement with FDA. Next slide, please. The measurable action within the combination of these molecules was identifying the cytokine storm that can be created within an inflammation scenario. The cytokine dysregulation that can occur through autoimmune disease, through even in oncology, dexamethasone is predominantly used as a corticosteroid prescription when there is a cytokine storm going in within the body, and therefore the cytokines that we focused on were all of them initially. There's pro-inflammatory and then there's anti-inflammatory cytokines. And so we focused and narrowed the focus on the pro-inflammatory cytokines as a way to preserve the anti-inflammatory cytokines so that we could lower the pro-inflammatory cytokine activity. And this is where the mechanism of action of these molecules and the results are quite impressive as it relates to just identifying what happens in a cytokine release storm or a cytokine release syndrome that some oncology medications create. There's bispecific antibodies in cancer treatments that dexamethasone is used as a kind of a blunt anti-inflammatory. We compared in all of our models this one-to-one cannabidiol and cannabidiolic acid to dexamethasone as the benchmark. And it's quite impressive, the combination, just from a layman's perspective. I'm not a scientist, so my terminology and language is just of wow factor when I am explained the scientific action, the mechanism of action. Next slide, please. We're currently on an IND track to build this asset out in an ability to license into the pharmaceutical arena. We're currently in a gap analysis right now, as previously mentioned, to identify our exact indication because there are so many indications as it relates to anti-inflammatory that we are narrowing that focus, and that is active. We will more than likely have our indication identified within this calendar month, and with the gap analysis, it will determine exactly what additional studies need to be done prior to an IND engagement. Our next stage will be the pre-IND meetings, which we suspect will happen within this calendar year, and then the IND filing, which will be in 2027. Next slide, please. So very clean, preclinically proven within a university relationship, patent protected within the methods of use, internationally protected. But what CitraChem allows for us to really narrow our focus on is really the scalability of a pharmaceutical grade molecule set that we can enter through the CMC controls, through FDA engagement confidently with the reproducibility, the scalability. There's just a lot that CitraChem affords us because, as mentioned, we're only going to be as strong as the chemistry behind it. And so we're very excited to see CitraChem succeed and move forward because it is very impressive because of the fewer steps necessary to identify and to secure those molecules. So very simply, this is not just a wellness story. We are really talking about a pharmaceutical asset and moving into FDA engagement. And then the final slide is the end of my presentation. So any questions, I'm happy to answer. And you can direct through James as well for post-presentation. But that is the conclusion of my presentation today.
Jeff, we really appreciate your time. I don't see any questions in the queue, but for the audience, you have been involved in a botanical over-the-counter product, and it was important to you to move to FDA application. You want to just explain a little bit why that was important?
So when you're working with intellectual property, you want to uh, really put the technology in play. You don't want to essentially just file IP for the sake of filing IP. And because of the lack of a regulatory framework for dietary supplements, we chose to use the botanically sourced molecules where we use two isolates derived from botanical sources because our IP also protects us on botanically sourced or synthetically sourced molecules. And over the past 7 years, we have been actively selling under a branded entity uh, through our own management of that entity from a business perspective. The feedback from consumers that consume these products, not only in the cosmetic form, but also in the dietary supplement form. And the feedback that we've received over the years is just game changing. We all, as a team, drink the Kool-Aid per se, meaning that we use the product and it is a noticeable difference in the sense of knowing when you don't take it or when you don't use it. You miss it. As an example, my before every round of golf, I will use our topical products, and I can gain better grip strength in my hands. About 45 minutes before a round of golf. Not that that's my cheat code, but ultimately, I can recognize when I don't prepare for a round of golf, my grip strength is not the same. So those are just little nuances that I've noticed just as anecdotal evidence as it relates. My 84-year-old mother has arthritis. She talks about how her fingernails grow stronger. But the biggest takeaway that we have, I think we've served over 70,000 customers over the past 7 years and continue to sell the products today that are botanically sourced. But it's really when uh, they run out of the product that they miss the product, and that is what stands out to me the most.
And what was that decision to then move to FDA track?
The intellectual property issuances, I think that the confidence that we have in the formulary, the regulatory environment of FDA, HHS activity, there is a lot more support of cannabinoids through pharmaceutical adoption. The actual indication in disease states, I think that the dexamethasone benchmark and where this compares as the one-to-one equimolar really stands out to where it should be within the pharmaceutical arena and not necessarily dietary supplement because we want to be able to say exactly what it does. And in the dietary supplement arena, we can't talk about inflammation or anti-inflammatory. We can only speak to the purity and the combination. Therefore, consumers do not understand why they need to take it or why they would want to take it. And that's why we have narrowed our focus on the FDA pathway.
Makes sense. Well, we appreciate your time again. Enjoy the rest of your visit here in New England.
Fantastic. Thank you so much. I will stay on for the final presentation and the summary. So thank you for having me on today.
You are welcome. Okay. So that is the main part of our presentation. As just a quick wrap up to summarize, we really wanted to demonstrate the significant work that's already happening in cannabinoid medicine research. Hopefully you saw from this set of presenters, we are collaborating with others as well. Really what that means is, part of my presentation was we have a platform. We want to provide ingredients to as many drug discovery companies as we can. You just heard from a few of those. Our mission is to develop relationships like these far and wide. And as we discussed at the beginning, there's so many ways to utilize these medicines, and there's so many possibilities coming out of the plant that we can either replicate or we can change and improve. It's nearly infinite. I like to say that. Someone might object to that, but I'm going to say it's nearly infinite, especially when you look at the map that I'm showing here. So CitraChem is prepared to go the next step. We are in the process of looking at our funding for getting more natural cannabinoids in our library and being able to communicate in the manner that we are today with more drug discovery companies, academic institutions that we can provide this work. Really appreciate you tuning in today. If you have any questions, please do not hesitate to contact me. You can contact any of our presenters directly, but we're happy to facilitate that as needed. Thank you very much, and that is the end of our presentation for today. We hope to do this again in the fall in September, and we'll keep you informed. Thank you.